Hello everyone, welcome to the channel Study with Asmita. Today we are going to start with the chapter number 1 of class 12, that is sexual reproduction in flowering plants. So in our previous class we have already classified all the living organisms on the basis of their similarities and dissimilarities. One of the hierarchical classification was kingdom. So plant kingdom was further classified into 5 divisions, which were thallophytes, bryophyte, pteridophyte, gymnosperm and angiosperms, right? So, so angiosperms are the most developed division of this kingdom as it bears flowers for its sexual reproduction. So, we are going to take a detailed look about its sexual reproduction in this chapter. Before starting the chapter, we are going to discuss about the topic which we have to cover in this chapter, which are flower, a fascinating organ of angiosperm, then pre-fertilization is structure and events, then double fertilization, post-fertilization, apomixis and polyembryony. Now let's start with our new topic, that is flower, which is basically a fascinating organ of angiosperm whose origin is axillary bud. It is basically an organ which is a site of sexual reproduction in flowering plants. Before starting with the chapter, let us take a look of all the parts of a flower. If we talk about pistil or the carpel, it is the female part of a plant which consists of stigma, style and ovary. If we talk about the stamen, it is the male part of a plant which consists of anther and a filament. Now other these reproductive parts, there are accessory parts which are petal, which is a colorful part of a plant which helps in attracting the insects for pollination. If we talk about sepal, it is a green leafy like structure which provide protection to the birds in its initial stage. If we talk about this receptacles or the thalamus, it is basically a swollen part of a flower. Now pedicle, it is basically a part where bird is joined to the stem or a branch. Now ring-like structure formed by stamen is known as androecium, and group of pistil or carpel is known as gynoecium. Calyx, it is a group of sepals, and group of petals are known as corolla. Now let's start with another topic, that is pre-fertilization, structure and events. Now in the case of male, which is a stamen in a flowering plant, and it is a unit of androecium. So stamen is basically the male part, as we have discussed earlier, which consists of anther and the filament. Inside the anther there is a connective middle tissue. Anther, it is bilobed, means it has 2 lobes, right? And each lobe here 2 theca. So this is the diagram of theca here from inside. Okay, here is microsporangium in each theca, which consists of pollen sacs, one in each. Okay, basically it forms haploid pollen grains, or we can say 2 male gametes. If we talk about filament, this is the stalk-like structure which provide exposure to the anther. Now this proximal part is attached to thalamus. Now if we talk about connective tissue, it is a site of attachment of filament with anther. Now let's take a look at the structure of microsporangium, which consists of 4 walls and diploid sporogenous tissue. If we talk about the 4 walls, then out of these 4 walls, 3 walls are protective walls, and the 4th one, that is tapetum, is the nutritive wall. So the epidermis, endothelium, middle layers are the protective walls. Okay, epidermis and endothelium are the single layer, and middle layers are basically multiple layered, which is just next to endothelium. Now if we talk about the tapetum, which contains dense cytoplasm and are multinucleated. Now if we talk about the diploid sporogenous tissue, okay, which forms MMC, that is microspore mother cell, okay, that is also, then it further develop into Pauline mother cell. By the process of meiosis, haploid microspores are formed, which further develops into Pauline grains, which are also known as male gametophyte, which is 2 in number. Okay, so let us take a look of diagram. This middle part is the connective tissue, then this outer layer is the epidermis. Next to epidermis, it is endothelium, which are both are single layered. Okay, next to the endothelium, multi layered, which consists of multiple layers. Okay, tapetum is next to the middle layers, which provide nutrition to the growing pollen grain. Okay, and next to the tapetum, sporogenous tissues are present. Now, if we talk about microsporogenesis, then it is the process of formation of haploid microspores from diploid microspores mother cell or the pollen mother cell, that is PMC, through meiotic division is called microsporogenesis. As we have seen, the sporogenous tissues present here, which consist of pollen mother cell, okay, in which meiotic division take place, and by the help of this division, haploid microspores are formed. This process is known as microsporogenesis. Then each microspore further develops to form mature pollen grain, that is male gametophyte. Now here in the diagram, the process of microsporogenesis is shown. Pollen grain represent the male gametophyte in the angiosperms. Okay. So the conclusion is pollen grain formed from microspores tetrad after microsporogenesis. Now coming to the topics, shedding of pollen grains on stigma. In 60% of angiosperm, pollen grain sets at two cell stage. One is generative and the other one is vegetative. Now in other 40% of angiosperm, pollen grain sets at three cell stage, that is one is vegetative, another two are two male gametes. Okay. Now we will talk about the harmful effects of the pollen grains. Pollen grains cause pollen allergy and also causes chronic respiratory disorder. The example of carrot grass, which is also known as parthenium or weed of weeds, which cause allergy, and in the case of respiratory disorder, it causes bronchitis and asthma. Now we are going to take a look of the importance of pollen grains. Okay, so pollen grains are basically rich in proteins. So in Western countries, it is taken as a food supplement in the form of tablets or syrups. Now if we talk about the viability of a pollen grain, it can be from 30 minutes to even several months and years. Right? For example, in the case of wheat and rye, its viability is just for 30 minutes, and in the families such as Rosaceae, Leguminosae and Solanaceae, it can viable for several months and several years. Now pollen grains can be stored in liquid nitrogen at minus 196 degrees Celsius. It is very useful in plant breeding programs. Now with the next topic we are going to start with is the pistil megasporangium, that is ovule, and embryo sac female gametophyte. Now here we are going to take a look of female parts and its function. Okay, so gynosium is basically is a group of carpel or pistil, okay, which consist of stigma, style, and ovary. So this is the stigma, this is upper flattened part, okay, this is the style, middle tubular part, and the ovary, this is the basal swollen part, which consists of one or more ovules. Now, in angiosperm, gynosium can be divided into two parts, that is monocarpellary, which the flower in which single carpel is present, okay, and another one is multicarpellary. It is further divided into two parts, that is fused carpels and free carpels. Or with the free carpels are known as apocarpus and fused carpels are known as syncarpus. Now we are going to talk about the ovary, okay, which consists of ovary wall and ovarian cavity. Inside the ovarian cavity, locule is present, which is further divided into placenta and ovule. Placenta basically it provide nutrition to the developing ovule. Ovule is also known as megasporangium. Now we'll talk about the ovule or megasporangium. This is our topic. So it is followed. So its structure is divided into two parts, that is external and the internal. Funicle is the stalk of the ovule attached to the placenta. Then coming to hilum, attachment site of ovule with the funicle. Now integuments are the covering of ovule, which is further divided into two parts, outer integuments and inner integuments. Okay, now the micropyle end is a place where integuments are absent, which is just opposite to chalazal pole. Now chalazal pole is a part of ovule opposite to micropyle. Now if we talk about internal structure, it consists of nucleus, inside which diploid cells provide nutrition to the developing embryo sac. So micropyle is basically opening for ovule for the entry of pollen tube. Now let us take a look of its diagram. This is the funicle, okay. Next to it, it is the hilum. This is the outer integuments and the inner integuments. You can see here that it is the opening, which is known as micropyle, so that pollen tube can enter from here, and chalazal pole is just opposite to micropylar pole. Now this is the outer integuments, inner integuments, and these are the nucleus, and this is megaspore mother cell. Now we will talk about megasporogenesis, which is the process of formation of haploid megaspores from diploid megaspore mother cell by meiotic division. Okay, haploid megaspore develop to form haploid female gametophyte, that is embryo sac. Now this is the diploid megaspore mother cell. Now we can see here the diploid megaspore mother cell, okay, inside which nucleus is present, okay, then it is followed by meiosis first. Now with the help of diagram we are going to understand the process of megasporogenesis, okay. So now in the nucleus of megaspore mother cell, meiosis division takes place. The first meiosis first result into division of the nuclei, okay, and it forms megaspore dyad. Then again it divides and forms four nucleus in it, okay, out of which three degenerates and only one remains, the functional. In the functional megaspore, mitosis takes place and it forms two nucleate stage. Again it divides and forms four nucleate stage, which is followed by mitosis again, in which 8 nucleus are formed, okay. So this is the 8 nucleate stage. Here the cytokinesis take place. Okay, at the micropylar end, filiform apparatus, synergid cell, okay, and egg cell are formed. Right between at the middle, polar nuclei remain. Okay, this is only the one cell, and two nuclei are present here. Now, at the chalazal end, antipodal cells are formed, which is three in number. Three at the chalazal end and three number of cells at the micropylar end, and two nucleus, one cell is formed at the center. So this stage is known as cell and eight nucleate stage. Now, formation of embryo sac from single functional megaspore is known as monosporic. As we have seen above, that other three megaspores are getting degenerated, right? And only one of these are functional megaspore. So such kind of development of embryo is known as monosporic development. Okay. Now, if we talk about female gametophyte, that is embryo sac, also 7-celled and 8 nucleated stage at. So, as we have seen in the previous slide, that is micropylar end and chalazal end. So, at the micropylar end, egg apparatus is formed, out of which two synergid cell and one egg cell is present. Okay, in the central part that has a central cell. Now in egg apparatus, two synergy cells are present at the egg apparatus. A filiform apparatus which guides the entry of the pollen tube, okay, during fertilization. Now if we talk about the egg cell, which is haploid female gamete, okay, and formation of diploid zygote after fertilization takes place here. Now in the case of central cell, two haploid polar nuclei are present. Formation of triploid endosperm after double fertilization and triple fusion takes place. Okay. If we talk about the chalazal end, three haploid antipodal cells are present whose function are not known till yet, and it degenerates after fertilization. Now we are going to start with the next topic, that is pollination, which can be defined as transfer of pollen grains from anther to the stigma. Okay, which can be further classified into two types, that is self-pollination and cross-pollination. This self-pollination is further classified into two types, that is autogamy and geitonogamy, and cross-pollination is known as genogamy. Okay, the first one is autogamy, that is transfer of pollen grains from anther to the stigma of the same plant. That means in this case autogamy takes place. Okay. Now, if we talk about the flowers, there are two types of flowers, that are chasmogamous and cristogamous flowers. Chasmogamous are the open flowers with exposed anther and stigma, whereas clistogamous are closed flowers. So coming to geitonogamy, transfer of pollen grain from anther of one flower to the stigma of another flower of the same plant is known as geitonogamy. Suppose, taking an example, this is a plant, this is the male flower, and here is the female flower. If the pollination is taken between these two flowers, it is said to be geitonogamy. Example of geitonogamy is cucumber. Now coming to genogamy, which is also known as cross-pollination, it is basically transfer of pollen grain from anther of one flower to the stigma of another flower that is born on two different plants. Suppose two different plants are here and the pollination is taking part between the flowers of these plants is known as cross-pollination. Now, pollinating agents can be abiotic agents or biotic agents. Now, abiotic agents can be wind or water. Wind pollination is known as aminophily, which is common in monocots like grass, whereas in case of water, it's known as hydrophily, which is common in hydrilla, seawater. Coming to biotic agents, which is mostly done by animals by different ways. One of them is insect, which is most common pollinating agents, which is done by bees and butterfly. Other creatures such as reptiles, example garden lizard and lizard are also a pollinating agent. In case of birds, sunbirds and hummingbirds are pollinating agents, and in case of mammals, bat also acts as a pollinating agent. Coming to the features of flowers pollinated by different biotic and abiotic agents. Okay, so the first one is wind pollinated flowers. So, in case of wind pollinated flowers, the pollen grains are non sticky and light. They have well exposed anther and feathery stigma. They are also odorless, nectarless, and colorless flowers. They, are, they have small flowers that are present in bunches. When it comes to water pollinated flowers, mucilaginous coat is present on the floral parts. The most important point here is pollen grain are of ribbon shape or ribbon like pollen grains are present. These flowers are also colorless, odorless, and nectarless flowers. Now, in case of insects pollinated flowers, these flowers have sticky pollen grains, attractive flowers with colorful petals, and flowers with edible pollen grains, and nectar is present at the base of the petals. Now, floral rewards for animals which are color, nectar, odor, and edible pollen grains, and the most important one is safe place for laying eggs. In case of Amorphophilous flower and yucca plant, the Amorphophilous flower is itself 6 feet height and which is a safe place for laying eggs. Okay, if we come up to yucca plants, then moth lay eggs in its ovarian cavity. An important note here, yucca and moth cannot complete their life cycle without each other. This is known as coexistence. Water is not a pollinating agent in all hydrophyte. Okay, in case of water hyacinth and water lily, we can see that it gets pollinated by insects or wind because their flowers are located above the water surface. Now we are going to see the advantages and the disadvantages of self and cross pollination. For the first, we are going to see the self pollination advantages, which are it maintains purity and there is no chance of mixing. Second, more chance of pollination. Then the third one is there is no need to produce large number of pollen grains. And the last one is no need of the floral rewards. Coming to disadvantages, it causes inbreeding depression, weaker progeny, and no variation, so it does not lead to evolution. Now, coming to cross pollination, the first one is advantages. So, advantages are higher yield, strong progeny. Progeny are highly resistant to several biotic and abiotic factors. And the last one is variation and evolution takes place. Disadvantages of cross pollination is appearance of some undesirable features along with desirable features, and the last one is uncertainty. Now coming to the topic outbreeding devices. in which plants have developed their own mechanism to prevent from inbreeding and to promote outbreeding. In case of monoecious plant, two types of flowers are there, bisexual and unisexual flowers. So in case of bisexual flowers, the time of release of pollen grain and stigma receptivity is different, so it generally protects autogamy. So it generally prevents autogamy, while the different way is the difference between the position of anther and the stigma. In case of unisexual flower, it prevents autogamy but not prevents geitonogamy. In case of dioecious plant, it prevents both autogamy and geitonogamy, as female flower is on different plant and male flower is on different plant. So basically this happens in the case of papaya. Coming to self-incompatibility, that is a genetic ability to recognize self-pollen grains and prevent pollen germination by the bisexual flowers. Pollen pistil interaction. So pollen pistil interaction includes all the events from landing of pollen grain on the stigma to the formation of pollen tube and entry of the pollen tube in one of the synergy cell of egg apparatus in the embryo sac. Now coming to the steps of pollen pistil interaction, which includes all the following steps. Now the first one is chemical interaction between the pollen grain and the stigma, as stigma should identify whether the pollen grain which has landed on the stigma is whether of the same species or not, right? Now here the pollen grain should be at 2-cell stage so that the further steps can be performed. Coming to third step, which is pollen tube formation. Now if all the things had happened in a right manner, then formation of pollen tube takes place through the style, and at the end the entry of pollen tube in the ovule of one of the synergy cell through micropyle and takes place. Now with the help of diagram we are going to understand this mechanism. Here the pollen grain, which is in 2-cell stage, is forming a pollen tube and reaching to one of the synergid cell, which is guided by pollen apparatus. Now we are going to start with the topic, that is artificial hybridization. Now the flowers has been classified into two types, right? That are bisexual flowers and unisexual flowers. In case of bisexual flowers, the male part, that is anther, has to be removed from the flower, that is known as emasculation. This process is known as emasculation, as the anther is removed from the flower. Now it is followed by bagging, in which the emasculated flower has to be covered with the butter paper so that any other pollen grain do not reach to the stigma. Now coming to the third step, that is artificial pollination. Now here the desirable pollen grain is dusted on the stigma after removing the butter paper. Now again the last step is rebagging. Once again the covering of a stigma with a desired pollen grain has to be done. Now if we come to unisexual flowers, as we all know that there is only one kind of reproductive parts in unisexual flowers, So there's no need of emasculation. We just need to do the bagging and then artificial pollination and rebagging in the similar manner with the bisexual flowers. Double fertilization, which is a unique feature of angiosperms, as the name of the topic suggests, double fertilization. So in angiosperms, two kinds of fertilization takes place, that is the generative and the other one is vegetative. In generative fertilization, basically this is a syngamy in which the fusion of haploid gametes with haploid egg takes place, which form zygote and further it develops into an embryo, while the other vegetative is a triple fusion. The fusion of haploid polar nuclei, which form diploid secondary nucleus, which is basically the fusion of another male gamete with the diploid secondary nucleus. Okay, so the formation of triploid primary endospermic nucleus, that is PEN, in the central cell takes place, and this is a nutritive tissue which provides nutrition to the developing embryo. This is known as endosperm. With the help of diagram, let us understand the process of double fertilization. So the two male gametes entering through the pollen tube in one of the synergid cell, right? And through the synergid cell, it reaches to the egg cell, and here syngamy takes place, which further develop as an embryo. Now, while the other reaches to the polar nuclei, which is already in the diploid state, and this result in triple fusion. So this is the zygote which is formed here through syngamy, and then this is the another male gamete which is reaching to the polar nuclei and form triploid nucleus, which is PEN, known as primary endosperm cell, which basically provide nutrition to the developing embryo. Now coming to the topic post-fertilization structure and events. So post-fertilization are the events after the fertilization in which endosperm formation takes place, okay, which is triploid tissue formed from triploid endosperm cell, which provide nutrition to the developing embryo, and then embryogenesis, which is the formation of embryo from the zygote, which can further be classified into monocot embryo or dicot embryo. After that, seed is formed from ovule, and then fruit is formed from ovary. So a key point is here, that is endosperm is a triploid tissue formed by the process of triple fusion during double fertilization. Okay, now let's talk about the types of endosperm, which can be classified into three types. The first one is nuclear endosperm, then the cellular, and then halobial. The nuclear endosperm is formed from the free nuclear division by the only process of karyokinesis in primary endosperm cell. Example, coconut water. Then the second one is cellular endosperm, which is formed by the karyokinesis in free nuclear endosperm, which leads to the formation of cellular endosperm, that is the white kernel edible part of the coconut. Then the third one is the intermediate of the nuclear and the cellular endosperm, which is mostly reported in monocots. Now, if we talk about the endosperm, there are two cases with the endosperm: whether to get completely consumed during the embryo development, or whether to remain even after embryo is developed. So, if the endosperm is remain in the seed that is known as endospermic seed. While if the endosperm has been completely consumed by the embryo, then that seed is known as non-endospermic seed. If we talk about the example of non-endospermic seeds are pea, gram, groundnuts and beans, while castor and coconut are the examples of endospermic seeds. Coming to embryo, and the formation of embryo is known as embryogenesis, which can be defined as the process of formation of diploid embryo from diploid zygote after fertilization. Okay. Initial development of embryo are similar in both monocot and dicots, but later on some differences occur separately in monocot and dicot embryos. So with the help of diagram, let us understand the process of embryogenesis in dicot embryo. So this is the zygote here, in which mitosis division taking place for the 3 times, right? And forming the cells. The basal one is the embryo at the 4 cell stage, and another 4 are basal cell, then the suspensor cell, and then hypophyses, which are aligned on each other on embryo at 4 cell stage, right? Then further development is taking place and it forms 3 suspensor diploid cell, and embryo reach at 8 cell stage. Here the peritoneal division takes place, and this is the vascular cell at the top. Then this is the suspensor cells. Then the embryo is more developed from the previous 8 celled stage. Then the further division and development forms the globular embryo. Now the development of globular embryo result in the heart-shaped embryo, which further develops and form mature dicot embryo. This is the suspensor cell, this is the plumule, this is the radicle, and these two are cotyledons. Now this is the diagram where we can see the hypocotyle, plumule and the cotyledons, radicle, and this is the root cap. Now coming to monocot embryo, development of monocot embryo is similar to dicot embryo up to the globular stage. In monocot, only single cotyledon is formed, which is at the one side of embryonal axis called scutellum. And in monocot embryo, both radicle is covered by coleorhiza and plumule is covered by coleoptile. The portion of embryonal axis above the level of attachment of scutellum is called epicotyle. So this is here the epicotyle, which is above the attachment of scutellum. Then epiblast is a small extension of embryo in monocots. Here it is the epiblast with a small extension. Coming to seed, it is the final product of the sexual reproduction in angiosperms. Seed is developed from ovule after fertilization. It is formed inside the ovarian cavity. Now the changes in ovule during the seed formation are: the first is diploid zygote, in which diploid embryo within the seed takes place, that is known as embryogenesis. Now coming to endosperm, which can be completely consumed during embryogenesis or may persist in the seed after embryogenesis. Now coming to nucleus, it may persist in some seed as thin layer called perisperm, for example in beet and black pepper. Now some changes occur in micropyle. Then coming to integuments, that are the seed coats. The first one is inner integument, that is the inner seed coat known as tegmen. Then the second one is outer integuments or the outer seed coat, that is known as testa. Coming to endosperm, if endosperm is consumed completely, then those seeds are known as non-albuminous seeds, while if it persists in some of the seeds, that seeds are known as albuminous seeds such as maize and wheat. Coming to seed dormancy, it is inactive phase of embryo within the seed due to slow metabolism, or we can also define it as period from seed formation to seed germination. So, after breaking the dormancy of seed, it germinates to produce a new plant under favorable conditions, right? It requires right amount of moisture, suitable temperature, and water and soil. So, this is the diagram of fertilized ovule with this 3N endosperm and the zygote 2N. Now this is a diagram of a gram with a germ pore and a seed coat. This is the germinating seed with plumule, the two cotyledons and the radicle. This is a monocot seed. This is the pericarp here, areolar layer, and the endosperm here. This is the radicle. Then here is the plumule, coleoptile just above the plumule, and scutellum here, and this is the root cap. So the cotyledons here in the seed basically provide the nutrition to the embryo during the dormancy. So dicot plants have two cotyledons, whereas monocot plants have single cotyledons. Fruit is the name of mature and ripened ovary after fertilization during transformation of ovules into the seeds. Okay, now the changes in ovary during the fruit formation are: first in ovary wall, that it forms fruit wall, that is pericarp, the epicarp and the mesocarp and the endocarp. Then the ovarian cavity, it is transformation of ovules into seeds. Now types of fruits we are going to look for. So the first one is true fruit, then parthenocarpic fruits, and then false fruit. The first true fruit are developed from the ovary, such as mango. Then in parthenocarpic fruit, the fruit is developed from the ovary without fertilization. These fruit are generally seedless fruits such as banana. Now coming to the false fruit, it developed from ovary along with other floral parts such as thalamus and sepals. In case of apple, it is grown from the thalamus part of the plant, and in case of strawberry, it has grown from sepals and thalamus. A note is given here that parthenocarpy can be induced by the application of PGR, that is plant growth regulator, like auxin, example in the case of papaya and tomato. Now we are going to look at the advantages of the seed formation in angiosperms. So Seeds is the basis of agriculture. It also contains baby plant inside it. It have better adaptive strategy for the dispersal so that plant species can colonize in new habitats. Dominant stage of few seeds have been formed which are very old viable seed. In the case of Lupinus articus, its dormant period is 10 years, and another example is for 2000 years is of a date palm. Now coming to the next topic, apomixis and polyembryony. Apomixis is basically a Greek word which have two parts, that is apo, which means without, and mixis means mixing, that is development of seeds without fertilization. So it is a kind of asexual reproduction which mimics the sexual reproduction in the flowering parts. So in the case of apomixis, a diploid egg mother cell is directly developed to form diploid embryo within the ovule without fertilization. Suppose this is the mother cell which directly develops into the embryo, okay, and it mimics the sexual reproduction. Now mostly it is reported in the family of Asteraceae and in some grasses. The condition where more than one embryos are formed within ovule during seed formation, for example citrus and mango. Now in such plants, some of the diploid nucleus cell develop to form multiple embryos without fertilization. Coming to importance of apomixis, apomixis seeds maintain their character over several generations and farmers do not have to buy hybrid seeds every year. It also helps in the biotechnology to find out the gene and the characters of apomatic gene to the various hybrid varieties of the seeds. With this, we came to the end of the video and the chapter as well. If there is any doubt, you can ask me in the comment section. Till then, take care of yourself. Keep on studying, keep on enjoying. Do like, share, subscribe to the channel. Do not forget to press the bell icon. And thanks for watching.